Hi everyone, my name is Laura Lubert and I'm also a postdoctoral fellow at the Eric and Wendy Schmidt Center, and it's my honor to present our next speaker, Professor Patrick Hsu. Professor Hsu is a co-founder and co-investigator at the Arc Institute, as well as an assistant professor at Berkeley University. His work has been pretty influential from the get-go. During his PhD, he played a pivotal role in developing CRISPR-Cas9 technology, and since then, his lab has led groundbreaking work in genome editing, amongst others, CRISPR technology that targets RNA, and the recent discovery of a whole new class of guide RNAs called bridge RNAs. Together with Brian he also developed EVO2, one of the largest biological foundation models. Unsurprisingly, his work has been awarded several accolades, including the NIH Early Independence Award, the Rainwater Prize for Innovative Early Career Scientists, and Forbes 30 Under 30. Please join me in welcoming Professor Patrick Hsu.
Awesome. Well, thanks so much, Caroline and the organizers, for having me. And it's a real joy and privilege to be here today and tell you about some of our work and trying to bring together the fields of genetics and machine learning. As Laura kindly introduced, I've been thinking about ways that we can manipulate, biological molecules directly inside of living cells for a long time. And over the last few years, we started to really turn our attention to how we could actually model these molecules as well. And so if we, go to the words of Frances Arnold, the original inventor of directed evolution, right? She says, for today, we can, for all practical purposes, rewrite and edit any sequence of DNA, but we cannot compose it. Maybe we can cut and paste these pieces from nature's compositions, but we don't know how to write the bars for a single enzymic passage. But she ends by saying, however, evolution does know how to do this. And so we started thinking in the lab, how can we decipher the language of evolution to try to compose genomic information? So these advances in AI, as we all know, offer this universal framework that leverage data and compute at scale to uncover higher order patterns in diverse domains, including natural language, vision, or even embodied intelligence, right? And of course, we're trying to apply this machine learning paradigm to biological molecules. And over the past few years, we've seen some really incredible breakthroughs for protein structure prediction, ligand docking, looking at the complex folds of RNA, and so on. And that's obviously been really wonderful, but the problem is, despite the immense complexity of even these molecules and these modeling paradigms so far, right, the complexity of these molecules is really dwarfed by the overall complexity of an entire cell, which is many orders of magnitude larger and more complex. And so the cell's structure and its function are really built by the diverse interactions between all of these different molecules, which is a staggering number of combinatorial possibilities at multiple levels of regulation. So in short, this is really, really complicated. The good news is that all of this molecular logic fundamentally is encoded within DNA. That's a single sequence that contains all of the information that is needed, arguably, to create an entire organism. So in fact, the effects of evolution are actually passed down throughout generations of life via these DNA mutations. So in principle, you might be able to learn something about function, right, or the functional adaptations to natural selection by looking at DNA alone. And so we asked this simple question: can we create a machine that tries to learn the evolutionary distribution of these sequences from whole genomes and learn something about the interactions of all these different molecules that creates an entire organism? One of the central challenges in doing this is that biology uses a different language. It's foreign and not very human interpretable. So on the left, you can see this schematic of this so-called central dogma of molecular biology, where DNA is transcribed to RNA, which is translated to proteins, right? But you'll immediately see two things on the right. The first is that these sequences are continuous and directional. And the second, from the bottom moving upward, there's a hierarchy of information flow that's embedded across the different modes of this central dogma, where they're predictably transformed into different types of sequences and codons. And so to try to understand this molecular language, you can try to port over the sort of autoregressive paradigm from natural language modeling here to DNA, where if you want to do next token prediction for a famous passage from the English literature, to be or not to B, we all intuitively know the next token. We've pre-trained on talking to each other, right? But if I asked you GTG, CATCT, would you know what the next nucleotide is, right? And to give you an intuition for why, at massive scales of text, this next token prediction paradigm can lead to general reasoning and intelligence, right? It's useful to look at a few examples. So if if I said in my free time, I like to code, right? Or banana, it's not banana, right? I'll learn something about grammar, or even world knowledge. The capital of Azerbaijan is Baku and not London, right? Or I'm standing in Cambridge and not in San Francisco, right? But you can get more complex things, even like spatial reasoning, right? Iroh went into the kitchen to make tea. Standing next to Iroh, Zuko pondered his destiny. He probably then left the kitchen and not the bathroom, right? And so, kind of at massive scales of text, you can start to break apart unstructured data to find patterns. And this is effectively what we try to do in biological research, right? We know and learn in high school that we have genotype that's linked or underpins phenotype in some way. And we try to use that to divulge mechanism, and phenotype is in turn selected in order to influence genotype, right? And so by looking at the next base or the next something in a biological sequence, you can start to learn pretty interesting and diverse things about complex biological properties. So an example would be if the next nucleotide in my genome is going to be a healthy nucleotide, or is going to give me a devastating genetic disorder, at the abstraction level of residues, is the next residue going to be a functional catalytic residue, or it's going to be some catalytic null that inactivates my molecular machine? Or it might be if this next nucleotide is going to disrupt a hairpin in RNA secondary structure that no longer forms the aptamer shape that is required to bring a cofactor in order to do transcriptional regulation. Or even at the abstraction level of genes, we know in prokaryotic genomes, genes that function together are clustered together in functional operons, and so mod A and mod B is probably followed by mod C and not the kind of unaffiliated DNA repair enzyme Rec A, right? And so we try to take these intuitions to train an initial model called EVO1. This is a close collaboration with my ARC colleague Brian He, where we've seen so far in the field lots of effort on creating task-specific, specialized models like ESM for protein language, or models for regulatory DNA or for RNA language in particular. However, all of these different components, because they're fundamentally encoded inside of the genome, right? By training a genome foundation model at long context, we might be able to learn all of these different elements, but importantly, all of their combinatorial interactions as well. So we trained this initial model at 7B, 131K token context, and show that the model is both multimodal because it can understand these different modalities of the central dogma, but that it's also multi-scale. And I'll give you a few examples. In addition to designing proteins, right, where you're simply auto-completing some residue sequence, you can also design things like CRISPR systems, which orchestrates a Cas9 protein, a molecular scissors, with a non-coding guide RNA, right? Because these are fundamentally encoded within the genome, EVO understands the coevolutionary relationship between these two different sequences and is able to make functional EVO-designed Cas9s that are able to cut DNA in the test tube or inside of a cell, right? That's a generation task at the not just molecular level, but at a molecular systems level. But you can also do predictions at the cellular or organismal scale. So one thing that we're able to do here is to efficiently classify essential from non-essential genes, something that's the territory of experimental gene essentiality CRISPR screens that take months to years in the lab. Here, we can replace that with forward passes of a neural network, right? Even at the genome scale, we can generate long sequences at a megabase that looks kind of like a prokaryotic genome. You'll see that there's dense coding architecture in both strands. There are genes that can be translated into proteins embedded within these long, unconstrained generations. They contain protein structures that map to the distribution of naturally observed alpha helices or beta sheets, and their ontologies map to fundamental functional ontologies. We can even see in these single generations all of the tRNAs that you need to support protein translation. But unfortunately, we see only some of the rRNAs that you would need to make the ribosomes that would then in turn make those proteins, right? So we sort of think of this as a blurry picture of life, right? And so hopefully more scaling could actually get us to sharper understanding and reasoning about genomic content. So in training this model, we uncovered so-called scaling laws for DNA, which is something that we've seen in the large language modeling space, which is that there's a very predictable improvement in model performance as you pour in more compute, more pre-training data, or more parameters into the model. We see that type of pattern here, implying that these are similar strategies that can be used to improve genomic model performance, right? And so EVA1 was trained on 300 billion tokens, tokenized a single nucleotide resolution of prokaryotic and phage genomes. So these are the small, sort of single-cellular organisms. And in the next study, we wanted to move to understand eukaryotic genomes like ours, right, which are of course much longer, contain introns and kind of non-coding space, and are much longer and more complex. With EVO2, we scaled to over 9 trillion tokens of DNA across all observable evolution, including bacteria, archaea, and all eukarya, and asked if we could extend from 131K context to a million token context, if we'd be able to learn more about these sort of kind of eukaryotic scales of biology, right? And so just to give you a sense of this, right, individual molecules are, of course, quite small, and with EVO1, we can fit in things like functional operons, right, in prokaryotic genomes, where you have multiple different elements that are coded together in cis sequence. But as you extend to eukaryotic genomes, the scale of entire yeast chromosomes, human TADs, or entire bacterial genomes, right, you can actually fit all of this information in context at a million token context, right? We scaled this model to over 40 billion parameters using a multi-convolutional hybrid architecture known as striped hyena. In EVO2, we developed a new architecture known as striped hyena 2 that, like EVO1, uses these kind of gated convolutions striped with 10% attention that's able to evaluate long context at near linear time, and with improvements in throughput of training. Right? And so, this is essentially how we can try to crunch, these massive pre-training databases and data mixes without the, kind of massive GPU farms that you have in, industrial AI labs. So that this is cool. Lots of architecture stuff. What does the model learn about biology? Right? And so, you can feed in mutations into the model and ask the model what's going on with that mutation, right? This is an entire field of computational biology known as variant effect prediction, right? And so I'll give you a more concrete example. There's an important gene in the genome known as BRCA1, right? It's known to be able to cause some breast and ovarian cancers, right? If you have a causal mutation, right, many patients might elect for a double mastectomy. If you have a benign mutation, you might just elect for an annual mammogram. The problem, though, is most people have what are known as variants of unknown significance, which is fancy, science jargon for we don't know what the hell is going on. The good news is, again, EVO has an opinion about your mutation, and if you feed in all the different known BRCA1 mutations derived from ClinVar, a gold standard NIH database, you can see that EVO is state-of-the-art in classifying non-coding mutations and their effects on pathogenicity, and it's nearly state-of-the-art for coding mutations as well. Intriguingly, EVO in training has only ever seen a single reference human genome, whereas AlphaMissense from DeepMind was trained explicitly on human genomic variation. So it's very interesting that it's actually able to learn to this degree of performance by seeing primate genomes, zebrafish, bacteriophage, C. elegans, and so on. And now we're trying to inject the model with explicit information on rare and common genetic variation to see if we can make a single generalist state-of-the-art model across both coding and non-coding mutations. These were all zero-shot predictions, but it turns out the embeddings of the model, its learned representations, have actually deeper information about biology. And in fact, by training a supervised classifier on top of the Evo embeddings, you can actually bring it to state-of-the-art performance, implying that simple kind of prompt engineering or steering the model in token space is only kind of one way to improve the genomic language model performance or insights. One of the common criticisms of these large language models is that they're black box, right? The models are learning something, but what are we, right? And so there's a subfield of ML called mechinterp, or mechanistic interpretability, where you can try to break down neural circuits from these large neural networks into observable parts, right? And so there's a classic example in the field from Anthropic, which makes the Claude series of models, where they've broken down Claude into these different semantic features, one of which famously corresponds to the concept, semantic concept of a Golden Gate Bridge, right? And you can actually see across a whole bunch of different languages, these semantic features light up in the same place. If you ask Claude, what is your physical form, it will sensibly tell you it doesn't have one, it's simply an AI. But if you clamped this Golden Gate Bridge feature to 10x its weight, it now becomes obsessed with the idea of bridges. I'm a famous suspension bridge. I'm the iconic bridge itself with its beautiful orange color and sweeping suspension cables, right? If you try to prompt it to ask it to do math, to ask it, what you should cook tonight for dinner, it will tell you you should cook the Golden Gate Bridge, right? And so you can try to take this type of paradigm of breaking down this model into human observable parts, which is called sparse autoencoders, where you can try to break down and interpret the model activations onto any language model, including Evo. I'll first sort of note that Evo has never been trained with any explicit biological information. It's unannotated, it's unsupervised. You've only asked it to do next nucleotide prediction. Intriguingly, when you lay out the E. coli genome here, because it's a circular genome, you can see a bunch of different semantic features lighting up at different locations across the E. coli genome. And what we found was that there was a particular semantic feature that corresponds to the idea of prophage sequences, right? And in fact, it's actually doing a really great job at finding the boundaries of these prophage sequences, right? And so what the sort of classic bioinformatic way to do this is to take all these different genes, blast them, and try to figure out which ones match to a phage origin, right? And those are tools like FASTER and others where you try to call the boundaries. But the model somehow has just learned this bottoms up, right? And in fact, these same prophage features fire in the spacers of CRISPR repeat arrays, right? Where in the process of CRISPR bacterial adaptive immunity, these fingerprints are captured from invading bacteriophage into these repeat arrays. And the model, again, has sort of managed to identify specifically these spacers as foreign. So if you zoom in on a different part of the genome, you can find features that fire on other things like open reading frames, intergenic regions, transfer RNAs, ribosomal RNAs, but not just coding and non-coding features, but also features like the secondary structure of proteins, because proteins are, of course, embedded in the genome. And so you can see features that fire selectively on alpha helices or beta sheets and, even in the context of kind of known protein-protein interactions embedded inside of these functional operons. So that's all prokaryotic genomes. But if you moved and looked at eukaryotic genomes, you can actually see in the human genome that in non-coding promoters upstream of a transcriptional start site, you can see that there are these motifs or semantic features that fire on these sequence motifs that correspond, surprisingly, to known transcription factor binding motifs, right? Suggesting that the model may have some inkling about gene regulation or binding. You can also find features that correspond to eukaryotic gene architecture, like coding sequences, untranslated regions, but not just the concept of exons, but also the leading and lagging edge of exons. So it's able to find those boundaries. This same feature can be used to look at the human genome but also the woolly mammoth genome, for example, which was not seen in pre-training, showing some sense of generality of these types of coding features. Mechinterp can also be used to look at computer code, right? And it can find errors in computer code, like mine is riddled with it, for example. You can also find errors in the genetic code. And there are features that seem to fire preferentially on frameshift mutations and not on, for example, synonymous mutations. And so we're very intrigued by the opportunities here to try to understand and mechanistically interpret something about how these language models are learning about biological features, not just for classic, well-known features like I've shown you so far, but potentially to discover heretofore unknown things about the genome. In addition to breaking down the model into these human interpretable parts, again, you can try to use it for generation. And we tested EVO in 3 different generation tasks to try to make mitochondrial genomes, whole bacterial genomes, or entire eukaryotic chromosomes. So if you wanted to make a mitochondrial genome, which of course we all have, right? You can see our version lined up here in the middle of the slide in a dashed box, right? Here are 6 EVO-generated mitochondrial genomes, and what you'll see is that it's not simply memorizing and regurgitating your mitochondrial genome. In fact, sometimes it'll kind of flip the orientation of genes or shift them around slightly, but importantly, it keeps all of the core genes that are in the mitochondrial genome, and it preserves all of the key protein-protein interactions, for example, the core subunits of complex 1 of this encoded mitochondrial electron transport chain. You can also generate things that look like entire bacterial genomes, like the minimal sort of species Mycoplasma genitalium. And so you can see with EVO1, right, and so here we're showing you in blue PFAM hits to the known sort of database, and then you can see that EVO1 generations were missing a lot of kind of these known proteins, but this actually kind of significantly improves with EVO2. And to me, this kind of recalls the improvements in scaling of these image generation models, where if you're trying to make this kangaroo in a hoodie with a welcome friends sign, sort of the early version of the model gives you something that looks like something on the street of San Francisco, right? The sign is kind of unintelligible. It's not that cool. It looks a little sad, right? And the hope is, right, with EVO 3 and EVO 4 and so on, right, we'll be able to go from these slightly sharper understandings of the genome to something that could potentially be synthesized and tested directly inside of living cells. I think you can already do so in a few-shot manner. The goal is to one day be able to do this more zero-shot. This extends to eukaryotic chromosomes as well, but one of the things that we're often asked with genome language models is, hey, this is cool, you can embed DNA, I guess, but what about epigenetics, and what about RNA? And so we try to kind of answer some of those questions, where you can take this generative model, a DNA model like EVO, and try to actually score its generations with a model like an Informer or Borzoi, which has been explicitly trained on things like DHS or ChIP-seq data. And you can basically score these EVO generations based on predicted chromatin accessibility, right? And so in this sort of design task, you can try to ask the model to conform to user-specified peaks, wide attack-seq peaks or narrow attack-seq peaks, for example. And by adding more compute into the model at generation time, what you can see is that the model over time, with more thinking, is able to try to generate sequences that fit these user design patterns with higher fidelity. Right? The fun thing is that you can encode information in these attack-seq peaks, like dots and dashes, and write Morse code messages like ARC or EVO2. And now we're installing these directly into the genomes of living cells and doing real attack-seq on them to see if we're able to make those actual patterns. I'll sort of, kind of motivate that I think this is actually very general, and it's not just, an epigenome structure scoring function, but it could be, really any generic sequence to function predictor that you hook on top of a generative model in order to be able to design, more complex representations of biology. We think of this as a foundational model because it enables this app store of different applications across different modalities of the central dogma all the way up to entire cellular and organismal scale tasks. We've shown a few examples of that in this study, and we hope the community will be able to kind of build many more. Right? This is a fully open source model, and by fully open source I mean the pre-training data, the pre-training infra, and all of the model weights are fully available. The code is on GitHub. The model is hosted on Hugging Face. We have a front end called Evo Designer, which you can use to prompt and interpret model outputs, blast sequences, do structure prediction of the embedded proteins, and so on. And so, we've been really delighted by the initial reception and lots of folks who are building with it. And I hope you guys do too. I'll just sort of end by saying, these models that learn, on massive scales of DNA are just the beginning, right? And so, we're very excited about ways that you can improve the quality of the model predictions and generations by, doing lab in the loop with experimental feedback. And our hope is that by taking the ability to compose DNA combined with the programmable recombinases that we reported from the lab last year, we'll be able to install, DNA directly inside of living cells that's been programmed at a higher level of abstraction than the sort of few bases that we're able to edit in the modern gene editing paradigm, something that we're really thinking of as genome design. So I'll just sort of end by saying, I think scientific discovery is often a search problem, right? As we all know, you kind of have to kind of guess and check, right? Where we have multiple scientific hypotheses and you need to recursively test them to see what's going on, right? The problem is that this is very confusing, right? And it kind of feels like when you're a kid and you look at the night sky and you're trying to find that constellation, where's the Big Dipper, and you have no idea. And what you really want is something that's much more like a co-pilot, like the Night Sky app, that could tell you where the constellations are across space and also at different points in time, right? This is really, kind of how biology is orchestrated across space and time. And so one of the things that is a flagship initiative of ours at ARC is to build these virtual cells that are able to model not just inputs of universal genome representations like I showed you today, but also inputs from the environment where genotype and the environmental representations collaborate to create better predictions of phenotype. We're doing this across a variety of different tasks, including curating public data with AI agents, where we recently released the world's largest repository of single-cell data by actually creating an agentic crawler over all of NCBI SRA, and taking kind of unstructured data sets that have been contributed across the community to generate over 400 million cells. We're also, in-house generating, perturbational data, and then, setting up the evals and benchmarks and novel algorithms to try to, kind of understand these cell representations. So, I'll sort of, just say I hope I've convinced you today that, in the community, we kind of have the pieces to try to close this conceptual Turing loop, right, which was first proposed in 1936 by the, Alan Turing, the computer scientist, where, if you had a machine that can read, kind of compute and then write back to tape, you could in principle implement any computable algorithm. And by digitizing biological states, thinking about it and writing it back into cells, right, we can start to close this analogous Turing loop for biology. This has been, really wonderful, series of collaborations across ARC with Brian He. Evo 1 was led by Eric Nguyen, Matt Durant, Brian Kang, Dhruva Kachrakar, David Lee, and Michael Pauly. And, many folks contributed to Evo 2 across ARC. our partner NVIDIA, university collaborators, and industry collaborators like Goodfire for our mechanistic interpretability work. So yeah, thanks so much. I'm delighted to take questions.
Thank you so much. Questions?
Thank you for a great talk. So I think a lot about what information that might be spatially encoded in biological system that might not be encoded by sequence, for example. So have you encountered any tasks where your model just cannot predict it, and you think that it's actually because you don't have that three-dimensional information of the fold of the DNA, for example? Or do you have any such examples where you think that you'd need to encode spatial information better in order to perform the task?
I think a lot, right? Thank you for that question. And I think, embedded in your question, no pun intended, is the question of what is a virtual cell, right? And, I think it's the word that's in the zeitgeist today, and lots of folks are talking about it, but it's actually very poorly defined, right? And I think with both EVO and EVO2, we had to work really hard to actually just invent evals from scratch. That would be quantitative for the ML and CompBio community, and also I think conceptually interesting to the wet lab community in a way that folks would feel that these are actually useful tasks that I would actually care about if there was no AI involved whatsoever, right? And I think we'll need to be able to do that for these single cell foundation models and also for the spatial ones, right? So, complex questions like, dendritic cell activation in the tumor microenvironment, right? Or some sort of quorum behavior are the kinds of things that I think single cell models alone are unlikely to capture accurately, and spatial data, of course, will be important. And I think, multi-scale models of biology, right? Biological scales span from atoms to ecosystems, right? And I think we'll have different modeling types or paradigms that will independently get developed and get unified over time. We have these all-atom diffusion models today that are trying to do structure and ligand docking. We have these sequence to function models, and then we have these ways of trying to, embed natural language and EHR information. And I think right now they're being developed somewhat independently. I think with EVO, what we saw was that there were protein and RNA and codon and whatsoever language models. We tried to collapse them into a single paradigm by modeling genomes alone. I think conceptually analogous ways of collapsing these other types of models over time will also be needed.
Thank you so much, Patrick, for the amazing talk. We unfortunately have to move on for the sake of time, but
thank you.
Let's thank Patrick again.